linear form. Okay, and this corresponds to what they call history lambda. And another region is the section that has highly branched points, which is called amorphous lambda. Okay, so this growth is basically just the arithmetic pattern between history and amorphous lambda. And so this is the biosynthesis of the starch. So starting from Calvin cycle, and you get the final product, the sugar final product from this process. And this product will be sequentially converted into ATP glucose, which is a substrate of starch. And there are a few enzymes, these three enzymes that catalyze these glucose units and put them together into the structure that we showed in the previous slide. So basically in this work, I will focus only at this reaction. So here we start from glucose units, right? And there are a few enzymes that will catalyze this glucose, put them together to form such an interesting pattern that we see here. So when I saw the first time I saw this structure, I was really puzzled because, I asked myself, how could it be possible that a few enzymes and a bunch of glucose units inside the cell, right? They just bump to each other randomly. How come they could give you such a structure, right? Okay, to give you an idea of how wonderful of this structure is, let's say you have Lego building box here. Everyone has played Lego before, right? So let's say if I represent those glucose units by these Lego units. If you give this Lego set to a monkey, let them play with this Lego set, what you would get would be something like this, like a random pattern of Lego set that you give to the monkey, right? But if you give the same Lego set to, say, an intelligent creature, like an alien, you might get something like this, a pattern, a non-random, highly ordered pattern of the structure, like someone says that pyramids were built by aliens. You heard about that? So I guess this is quite an interesting question. How come you have to start with glucose units, right? And then you put 3 or 4 enzymes, and then they bump each other randomly, and then you get such a nice structure of starch. So the motivation of my research is that I want to explore the mechanisms underlying the distinct structure of amylopectin molecule. Okay? And to do this, I will use computational approach to study the interplays between key enzymes involved in this process. And the ultimate goal, if we really understand the underlying mechanism of the starch synthesis, then it is possible that we can use this knowledge to modify the structure of the starch, right? For example, we can increase the use for human and animal feeds. In some industry, they want to modify the starch property to have the property of glue or adhesive agents. We can even modify starch to be biodegradable packaging material, something like that. So first, let me introduce you to properties of enzymes that involved in this process. So we have three major groups of enzymes. The first one is starch synthesis, which I just call SS for short. And basically this enzyme will just add one glucose unit into the end of the existing chain of the molecule. Okay? So if you already have the chain of the amylopectin molecule, then SS will add one unit at the end of the chain. Okay? The second enzyme is branching enzymes, and I call it BE. This enzyme will cut part of the chain and then add that part into another chain, showing like shown here. Okay, so this one cut these two glucose units from one chain and put it into another chain. But you can see the difference between these two enzymes. The first enzyme put glucose unit in this position, right, which is actually the carbon position 4 of the chain. But BE will put the glucose units to carbon position 6, forming the branch point of the molecule. Okay. So basically this one is the extension process of the chain, and this one is the branching process of the chain. Okay. And the last enzyme is the branching enzyme, or the BE. This one is basically similar. It is like the reversing process of BE. It will cut. It will look for the branch points and then cut that branch. Okay. Showing this in the last panel. So the next thing is that I want to mimic the behavior of each enzymes that I just showed you in the previous slide. So first I want to represent glucose units by only just this blue dot, okay, for simplicity. Okay? And then I have another assumption that the growth of the molecule occurs in 2D. Okay? This is a big major assumption that I use for now for the simplicity of the model. And I just use green color to represent, to distinguish the bond point from other linear glucose units. Okay, but they are essentially the same. They all are the just glucose units. Okay, so this is for starch synthase, for SS. There are a couple more assumptions that, so say this is the space inside the cell, inside chloroplast or myeloplast, right? I assume that all enzymes, including SS, are abundant and they are everywhere inside this space. Okay? And we also have a lot of glucose units inside this space. Okay? So whenever you see a chain of the molecule, then SS will put a glucose unit at the end of the chain. Okay? So if I let the simulation begin, you will see just like that. SS will just add glucose units one at a time at the end of the chain. So this is the simulation for SS. And for BE, so for BE, I just assume that a chain that has the length longer than a specific value. So in this case, in this particular simulation is 12. So if a chain has the length longer than 12 glucose units, it will be cut by 6 units and it will add to its own chain like in this simulation. Okay. And DBE is just removing a bunch point from the molecule. So now you might wonder that if I have two chains, right, in this one, how do I choose which chain will be extended first and which chain will be extended by SS second, right? And that's why I have to use what we call stochastic modeling. Okay? So put it in a simple way, it's just that you have to list all possible reactions in your system, okay? And line those down in pieces of paper. So you have two chains. Both of them can be categorized by SS, right? Can be extended by SS. So I write down chain 1 in SS to represent the possible reaction for this chain, and chain 2 SS for also for the possible reaction for this chain, right? And then I just put these pieces of paper in the box, check it, and then randomly pick one of them. Okay. So if I randomly pick one paper, and that is this piece of paper, and I just add one glucose units to chain 1. Or if I pick another piece of paper, I will add the glucose unit to chain 2. Okay? And by repeating this process, you can do the stochastic simulation, something like this. So it will randomly add glucose units to your amylopectin molecule. And this is essentially what happens inside the cell, because inside the cell, enzymes and substrate are just randomly bound to each other, right? It's just the probability of the events that occur inside the cell. So now I just combine two enzymes together, SS and DE. Okay? Same thing. Now you have to check for each chain and see. Say if chain 1 can be categorized by SS, write it down in a piece of paper. If it can be categorized by DE, write it down in another piece of paper, and then just randomly pick one of them. Okay? And by repeating this process for 500 steps, I will get something like this, which is not exactly what we expected in the first place, because what we expected is to get something like this. This is the structure of amylopectin molecule, right? So you see that the main difference between these two structures is that in amylopectin molecule, the branch points are clustered together in layers, right? You see a lot of branch points here and then here, right? But in what I have here, you see that the branch points are scattered all over the place, right? All over the molecule. Okay, so this is not what I really expected for. Okay, but because now we have only two enzymes, right? Just SS and DE. So, and if you go to literature and see some papers that they study the loads of DBE, you will see that if you knock out this gene, the branch enzyme, if you knock out this gene from plants, then the structure of amylopectin will be converted into another abnormal structure called fibroglycogen. Okay, and we see that the pattern of the branch points in this molecule is random. It's not in order like in the amylopectin molecule.